Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I'm the marketing manager at Medicinal Genomics and proud member of the team that puts on the CanMed conference. So, if you have signed up for email alerts at canmedevents.com, or if you follow us on social media, then you saw that the CanMed 24 Innovation and Investment Summit will return to the JW Marriott Resort in Marco Island, Florida, May 12th through 15th. CanMed 23 was our first year on Marco Island, and the feedback was overwhelmingly positive. The beach, the food, the amenities, the networking opportunities, and of course, the content presented were all exceptional. And CanMed 24 is shaping up to be even better. Registration will be opening up soon, and tickets are limited. So if you haven't yet, sign up for email alerts at canmedevents.com so you are among the first to know when tickets go on sale. All right, so this episode we brought back Dr. Zamir Punja to talk about total yeast and mold. Zamir is a professor of plant biotechnology at Simon Fraser University in Canada. His research interests include the etiology and management of plant diseases on vegetable and horticultural crops, and the application of plant biotechnology for disease management. Since 2018, his work has shifted on researching cannabis, and his group has described a range of previously unreported pathogens affecting the crop, and has evaluated various methods for disease management. His team just published the results of a 3-year study that identified several factors that have a direct effect on yeast and mold levels. They include factors related to growing, drying, and trimming cannabis specifically. This is important because when growers fail for microbial testing, yeast and mold is often the reason. So if you are a grower that has struggled with total yeast and mold, perhaps these findings in the paper and on this podcast can help you get back on track. I've put a link to the paper in the show description, and if you're watching on YouTube, I've added several figures and images from the paper throughout the conversation. We cover the different species of yeast and mold that are commonly found on cannabis and which are potentially harmful to humans, the value of total yeast and mold testing as an indicator of product safety and quality, what is a reasonable threshold for total yeast and mold, how cannabis genotype affects total yeast and mold levels, why leaf litter drives up TYM levels, why hang drying is a better method than wet trimming, the drawbacks of irradiation, and more. Before we get to my conversation with Zamir, I want to thank this episode's sponsor, Agilent Technologies. Whether you manufacture or test cannabis, you face the challenge of delivering high-quality products, increasing sample volume, and meeting emerging regulations. Agilent offers best-in-class cannabis and hemp analysis solutions featuring robust instruments, software, services, and consumables. Their team of experts can develop, implement, and optimize methods to get you up and running quickly. Learn more at agilent.com. Okay, and without any further ado, please enjoy my conversation with Dr. Zamir Punja. Good morning, Zamir. Thanks again for joining us on the podcast.
Hi, Ben. Good to see you again.
Yeah, I know we had to get you back in. We had to get you back in quick too, because I know that you and your team, you recently published a great paper about total yeast and mold in cannabis. And that's one of our favorite topics here on the podcast and at Medicinal Genomics, as you know. Total yeast and mold seems to be one of those tests that growers really struggle with. And more often than not, if a batch is failing for microbial, it's either for total yeast and mold or maybe more specifically Aspergillus, which is a fungi and would be kind of included in that category. So Always good to explore that topic. And I know that your paper, it includes some factors that you've identified for causing high TYM levels. So hopefully we can get some good best practices or recommendations for growers that they can enact so that they have fewer failures. But before we get into that, I wanted to ask you why you and your team were interested in investigating TYM in cannabis.
Right. So, TYM in cannabis is certainly one of the most common things that growers will encounter. It's everywhere in the sense that yeast and mold is not specific to cannabis. Apples, blueberries, raspberries, strawberries, you name it, peanuts, any product that's grown out either in the field or even indoors will always capture a certain proportion of yeast and mold because these organisms feed off of plant material. So any exudates, any nutrients, anything coming out of a flower or a potato or a pod will attract some level of yeast and mold. For the most part, these yeasts and molds aren't necessarily harmful to people. When you consume an apple that's got some yeast and mold, the tendency is for the body to break it all down and digest it all, and it's basically gone. Cannabis, of course, because of the various ways it's used, whether it's inhaled or vaped or extracted from, yeast and mold apparently has raised greater concern in cannabis than it has potentially on other food crops. And so, of course, the regulatory agencies have established criteria for testing for yeast and mold and established levels that will cause a product to fail. So the reason it's hard to manage and the reason we were interested in it is because, it is everywhere. And we wanted to find out, are there certain things that growers may be doing or not doing that may increase or decrease the levels of yeast and mold in their product? So we didn't actually manipulate the yeast and molds that were naturally occurring, but what we did was we tried to understand if you planted a certain genotype, if you had a certain type of air circulation, if you were leaving a lot of leaf litter on the floor, would those things increase or decrease the levels of yeast and mold? And up until now, nobody's actually looked at that. We've looked at yeast and mold after a product is harvested. So, for example, if you irradiate it, what happens? If you heat it up to a certain temperature, what happens? If you analyze it in the lab using different methods, what happens? We wanted to find out from a grower perspective, what could they be doing to try to reduce those levels in flower?
Yeah, excellent. And so what did you find? I mean, and
Well,
more specifically, I know that you've identified like certain types of species that you found in more abundance than others. I don't know if that might be a good thing to explore.
right. So we sort of analyzed all the types of yeast and mold, we found a total, and we actually went a step further and identified all the different species using molecular methods. And penicillium was the one that seemed to be the most prevalent on cannabis flower. Now, again, penicillium is everywhere. You can find it indoors. If you've got a home that has slightly damp conditions, you'll find it in your basement. You'll find it in your rug. You'll find it sometimes in your clothes. I mean, it's there, and obviously most of us can deal with it, in our bodies because we're used to it. We may be inhaling it. It's not so much of an issue. But in cannabis, and we don't know enough about penicillium actually, it may or may not be of concern. So that was the most prevalent one we found. And then we found a bunch of others that normally occur in the air, things like alternaria and another fungus called cladosporium, which is quite common in the air. Aspergillus wasn't that high. The overall levels of Aspergillus we found wasn't high. Now, this is under greenhouse cultivation, right? So, we don't have a lot of plant material laying around. We're not next to a wooded area or a forest where you may have a lot of Aspergillus flying in, blowing in through the air. So that wasn't a big concern in terms of the sampling we did in the greenhouse. We found primarily the penicilliums and then some alternaria and cladosporium species present.
Yeah, no, I'm glad you mentioned Aspergillus. And were you looking specifically at the species level too? Like, where's it sort of the pathogenic strains of Aspergillus, your Flavus fumigatus, Niger terrius?
Yeah, so the primers we used should have been able to separate those out, because then we also looked at sequences. We didn't find a lot of the fumigatus. We didn't find a lot of the pathogenic strains. The one we found was called Aspergillus ochraceus, and it's actually a yellow Aspergillus. You know, sometimes you can, on a petri dish, when you grow these molds like we do for a long time, one of them is black, another one's green, another one's gray. Ours is yellow, and it's a species that is not on the current list of regulated Aspergillus species. Now, that's not to say it's not important. It's just for some reason it's never made it onto that list, and maybe it's not pathogenic, right? It hasn't been flagged as being a pathogenic species, but because these Aspergillus, they're pretty much everywhere. They're on decaying plant material, they're in the air. It just so happens that in the greenhouse, we picked up more of this yellow Aspergillus species. The second one we found was Aspergillus niger, which is a black Aspergillus. And again, that's not one of the really species of concern. The ones like the fumigatus and the other ones, we didn't see a lot of in the greenhouses that we were working with.
Yeah. No, Niger actually is on the list for many states as one of the big four pathogenics.
Okay. So we found a few of those. Yeah.
Interesting.
Yeah.
Any of the others that you've mentioned, are they known to be pathogenic or even benign to people? Like what are we looking at there?
So, you know, the biggest concern I would say with a lot of these yeast and molds is the toxins. And when I say toxin, I don't mean something really scary. Just they produce these chemical byproducts as they grow. If you grow it up in the lab, as these cultures grow, as they get a little bit older, they produce these chemical structures that are referred to as mycotoxins. Both Penicillium and Aspergillus will produce some of these mycotoxins, which, again, it's on the radar for testing. OTA, for example, ochratoxin, is a mycotoxin produced by both Aspergillus and Penicillium species. And it can cause some issues if it's ingested at a very high concentration. So, for example, you know, Penicillium, you see that on moldy cheese. Cheese that turns blue has Penicillium on it. If you ate a lot of that, you would probably end up with some issues in your tummy because, you know, there's secondary metabolites or toxins produced. We don't know how much or if any toxins are produced by the Penicillium species in cannabis. That's something that perhaps will end up on the radar eventually, how much of these toxins are produced. Fusarium, on the other hand, which is known to infect wheat and barley and other agricultural crops, does produce a lot of very dangerous mycotoxins. One of them is called vomitoxin, which when it's ingested, when the barley or wheat is ingested by animals like by cows or cattle, they'll vomit, like they'll throw up because there's so much of it in the grain. We probably, as humans, wouldn't be exposed to as much. But there are cases where poisoning has occurred by Fusarium mycotoxins. But again, we know nothing about what they may be doing or if they're even present in cannabis. The whole focus has been primarily on Aspergillus, for better or for worse. I mean, that's what the regulatory agencies decided was a priority.
Yeah, I mean, and I know that we've written about it before. It's been, I mean, it's one of the tests that we sell too, of course. But it is one of the few, if not the only, you know, contaminants on cannabis that has been shown to be actually fatal. So maybe for good reason that we're testing for Aspergillus.
Yeah, and that's the one that's definitely the most well known. You know, it causes the Aspergillosis in lungs of humans, farmers. It's sometimes referred to as farmers' lung disease because when farmers work with a lot of hay, with a lot of barley, wheat, over years they develop a sensitivity to the Aspergillus spores. And certainly people that are immunocompromised or have, you know, health conditions, sometimes diabetes, or even have had organ transplants, they will be more susceptible to problems with Aspergillus. But for the most part, you know, healthy individuals, and I would consider myself healthy, and you look pretty healthy to me. I doubt either one of us would suffer from Aspergillosis unless we were smoking, you know, 12, 14 joints a day that had Aspergillus in it. on a regular basis, we probably wouldn't suffer from this unless, we had some underlying conditions. But as you said, it can cause problems in humans. And so if there's any doubt, any concern, it is flagged. And so Aspergillus is flagged.
Right. And now when you were talking about, different species and everything, two that kind of stood out to me that maybe you didn't mention, or maybe you didn't even find: powdery mildew and botrytis.
Yeah.
Did you find that?
So powdery mildew is there, and you've seen it on buds. I've seen it on buds. It has this, white sort of a moldy appearance. Because we were growing these on culture media plates in the lab, that one doesn't actually grow on the plate, and so it is actually, fortunate or unfortunate, not on the radar for total yeast and mold testing, because you can't test it, you can't grow it. So you may see a mold, a bud that's almost white, covered in powdery mildew. It may pass because it doesn't grow. Botrytis was an interesting one. We don't see a lot of it growing on yeast and mold plates. We know it's there. I mean, you can see botrytis mold,
Sure.
but for some reason it's overgrown by some of these other fungi, which in some ways sort of leads to me to question some of these yeast and mold testing assays that are done from a cultural perspective. How do you know when you do a yeast and mold test that you're culturing everything that's important from a disease perspective? You may be missing, or you meaning the person doing the test may be missing certain species of fungi. And I think botrytis is one that's being missed. For some reason, it doesn't seem to want to grow as much. Now, does botrytis cause problems in humans? We don't know for sure. It does produce some of these metabolites, these mycotoxins that I referred to earlier. But again, it's not on the radar for testing from a regulatory perspective. So it skips. It's passed. Unless it's super moldy, as you know. I mean, if it's that bad, you don't want to sell that product anyway.
Sort of the moldy strawberry analogy that we always talk about. If you're in the grocery store and you see strawberries that are covered in mold, you're not going to just say, well, I guess it passed testing, so I'm going to buy it. You're going to go on to a different package.
And it's interesting that the biggest mold on strawberry is botrytis.
Interesting.
Yeah, that's the one that tends to cause strawberries to go bad if you haven't eaten them in a couple of days. Is botrytis. So we've probably been swallowing botrytis spores for a very long period of time.
Wow. And maybe that's a good thing to bring up too. In terms of like a total yeast and mold test, is it really kind of to protect the consumer from potentially getting sick, or is it more of just like a check on just good sanitation or hygienic processes that are going on in the grow?
Yeah, so I think it's both. The reason it's flagged from a regulatory or health perspective is because of the fact that there are these reports from the 70s and even 80s of aspergillosis occurring, or Aspergillus infections, sometimes in patients that have smoked cannabis. So there's been a correlation there. Now, how often is it? I don't know. Is it 1 in 100,000? Is it 1 in 10,000? We don't have those numbers to say that out of all the population of cannabis consumers, a certain proportion will get Aspergillus infections. But I guess if one person gets it, that's enough to raise a flag, a radar. The other yeasts and molds that are in there that you and I are consuming every day in our diet, whether it's a strawberry or blueberry or some other fresh fruit, our bodies deal with it and break it down, and it's not an issue. In cannabis, we really don't know. When we do these yeast and mold tests, and somebody comes to me and says, I got 20,000 or I got 40,000, I don't know what that means from a health perspective. And I don't think the health practitioners know what that means either. Certainly, the more it is, the more the chances of getting a problem. But if you told me that 20,000 is twice as bad as 10,000, I wouldn't believe you, because there is no data to support that. Now, if it's 100,000, I would say, boy, that's a lot. Like, you shouldn't have 100,000 CFUs in a gram. When we put that on a petri dish, that petri dish is covered. You can't see the petri dish. It's just got so much mold on it. To me, that's not a great product to sell. But if you told me it's 40,000 versus 10, I have no idea what that means. Now, in Canada, we've set the limit at 50, and so 50 seems to be a doable number. It's a reasonable number in that if you're below 50, you probably have a product that's other than the Aspergillus species, a product that's doable in a sense. The body can deal with that level of mold. If it's less than that, so much the better, because it's getting closer and closer to being almost clean. So different states have set up different numbers. I think Massachusetts is probably up around 100,000. Others maybe less. Countries like Israel, where if you're using cannabis for medical purposes, it's 0. Like the product has to be essentially irradiated to make sure it's totally clean. So we don't have data on what these numbers actually mean in terms of a health perspective.
Yeah. No, and I think that's interesting that in Canada it's set at 50, because like you mentioned, here in the States, I mean, it ranges. Iowa, I believe, if you look on the map that we have on our website, Iowa has it set at 10, literally 10 CFU, which seems almost impossible. And then you have Florida, and I believe Michigan, and maybe Connecticut too. They're up to 100,000, and everywhere in between. But it's all orders of 10. So it's funny that Canada is like, well, 50. So that's...
Well, we've always been known as a country that sort of tries to balance things out and make sure that we're approaching things from a reasonable perspective. I have never seen a cannabis flower come in at 0.
Yeah.
And I would never have seen a flower come in at 10. So that's unreasonable to me, to have something that low. Walking down the park to your car, you've already inhaled more than 10 colony forming units of mold because of the mold in the trees and in the flowers and everything else around you. So there has to be a reasonable number that says, I'm not saying 50 is reasonable, but there has to be a number that says, okay, we can deal with that from a grower perspective. When we see 50,000 on flower, it's usually a product that has been grown under very high humidity, or it's during the peak of summer where there's a lot of heat, or it's a flower that's had some damage done to it, because 50 is still a very high number to meet. In other words, most things should pass 50,000. I've seen maybe 2 to 3% of product get rejected because it's been over 50,000. So that's a good number to say that, okay, you know what, below 50, things are probably going to be okay. So 10 or 50 or 100 CFUs to me is very low. I mean, it's impossible, really, because everything out there has mold on it.
Right. And really, I mean, to kind of go back to what we were saying before, it's not so much how much mold, although that can be an indicator, but it's what type is on there too, right? Because, I mean,
yes.
So say you set the limit at 50, like you have in Canada. If you had 40,000 CFUs of, say, Aspergillus, that's much more concerning than if it were, one of these other non-pathogenic species.
Yes, I agree. And so in my opinion, there are maybe three groups of fungi that should be tested for. Let's say we decided we don't want to do total CFUs anymore, and we abandoned that approach and said, all right, we're just going to target certain fungi that we know historically have had an importance for humans and could potentially cause problems if they're present in flower or on product. One would definitely be Aspergillus, again, because of the historical significance of Aspergillus. I don't think anybody would argue that we should just exclude it. That would be turning a blind eye to reports of Aspergillus causing disease. The other is Fusarium. We know there's Fusarium on cannabis and hemp. We know Fusarium causes major problems on cereal and other crops because of the toxins. And then the third is Penicillium. Now, Penicillium is a tough one because there's so much of it, and not all of it is bad. Like I mentioned, the cheese, right? You've got Penicillium on your cheese. If you eat a little bit of moldy cheese, you're going to be fine. So, but if we are going to target 3 organisms, those would be the 3. The rest of it, just ignore it. Like we're seeing those every day in our diets. It hasn't caused a problem everywhere. I can find 30 to 40 different species of yeast and mold on any given cannabis flower, out of which maybe 2 is Aspergillus and 1 might be Fusarium. So really, Out of that list of 40, I would only be concerned with those three. I think we should just test for those three and let the others be as they may. Because a lot of times growers use beneficial products like trichoderma and
Right.
and Gliocladium and other fungi that are registered. They're allowed to be used on cannabis. They can cause yeast and mold numbers so high that the product fails. So you're coming to me now and saying, I've treated this product with trichoderma. I haven't used any pesticides. This product is clean, but it's failing because it's got huge numbers of spores of trichoderma, which has been found to be okay for people to have. And so that's a dilemma if we're testing just total yeast and mold. If we're testing for Aspergillus, Fusarium, Penicillium, the trichoderma one's going to pass.
Yeah. And now I know in the case of Aspergillus, probably because I'm more familiar with it, that, it likely, the regulation wouldn't be just any form of Aspergillus because there's all sorts of different species. It would be those 4 pathogenic ones that we had mentioned before. Is it the same case with Penicillium and Fusarium that
Yes.
there's, certain
I would
pathogenic ones?
say I would say there's like Penicillium citrinum is one species that's very, very common that produces a number of these mycotoxins. And certainly in Fusarium, there's a couple of 3 species. Graminearum is definitely one of them, and Pseudograminearum, Sporotrochioides. I'm just throwing these names out because these are names that we know about. So yes, for sure, you wouldn't necessarily want to go after every single Fusarium that may be there.
Right.
It would be targeted towards the ones that we know. So really, you're looking at about 3 and 3, 6, maybe 8 or 9 specific fungi that are known to be harmful to humans that have produced toxins, and we should monitor for those. And the others, obviously, and then maybe set a limit of, let's say, 50,000 total CFUs if we were going to use that.
Yeah.
And anything below that, don't worry about it. But then test for these 7 or 8, like we do Salmonella and E. coli and so on. We don't test for every bacterium that's present.
Well, in some states they do.
In some states they do. That's right. They have the total bacterial loads as well. Yeah.
Right. Yeah. But luckily, like you said, those thresholds are usually set pretty high, and that isn't one that growers seem to struggle with. But total yeast and mold is. And we did say that we were going to talk about some of the factors that your team identified. So let's get into that. And I think one of the major ones that you identified that was in this paper was that the plant genetics themselves play a huge role in the TYM levels. I wonder if you could speak a bit to that. And is it specifically like the physical structure of the plant in the flowers that leads to that, or is there something else going on? Like, does it have something to do with immunity?
Yeah. So that was a bit of a surprise to us as well, and I'm sort of glad we really included like 7 or 8 different types of genetics or genotypes in the study. Obviously, we were grabbing whatever we could, whatever was being grown at the time. So it wasn't a planned experiment, but we did find definite differences between the types of cannabis that were being grown. Primarily, it's to do with the morphology and the size of the flower structure. So the larger the flowers, the bigger the mass, if you wish, or volume, the higher the yeast and mold in general. Plants that produce very small buds, smaller buds, many of them, but smaller in size, not as dense, seem to have less of a yeast and mold. So there was definitely this physical correlation. In some cases, I do think there were chemicals produced in the flower that would be inhibiting yeast and mold. I say that because some of these flowers were, the yeast and mold was so low that it was almost impossible to explain just from the fact that the flowers were small. I think they were secreting some kind of a chemical product, a natural chemical product that was preventing these yeast and molds from growing. If we knew what those were, boy, we'd be off to a really good start from a research perspective. I mean, definitely that's something that needs to be looked at. I don't think it's something like terpenes or some of these other volatiles that we know. It's definitely not THC or CBD, because all of these were producing THC, CBD. It's something else that perhaps isn't on the radar yet, something that other researchers or breeders may want to look at. But certainly flower structure, flower size. And the reason the larger flowers seem to have more yeast and mold is the humidity, the moisture in those flowers is higher by 3 to 4% in terms of relative humidity than the outside air. And their temperature builds up in the flower. It's almost a mini greenhouse sitting on a plant, because it traps the solar radiation and it warms up. The buds warm up 2-3 degrees higher than the environment. So that humidity and moisture, the temperature, creates this little greenhouse that allows these yeasts and molds to build up. So one thing we asked ourselves, well, if you circulate the air, you turn a fan on close to the plants and let this air infiltrate through the flowers, what would happen? And boy, the yeast and molds dropped by 90%.
Oh, wow.
Now, you can't go out and put a fan next to every plant, but certainly you increase your airflow, your air movement, the humidity drops, the temperature drops, and your flowers have way less yeast and mold. And it's not impractical. In some greenhouses, you can set up 8 or 10 fans blowing in a certain direction, and that will really reduce the yeast and mold.
Yeah. And, I imagine growers too would be encouraged to hear you say that there could be something more than just the morphology of the buds that's contributing to it. Because if you think about it, growers want big, dense flowers, and to have that be associated with higher TYM, well, that's not good news at all.
No, certainly. And so if the chemicals are discovered, which they will be eventually, that will be very, very useful, because that's a natural way for the plant to protect itself. Now, plants don't have an immune system, but they do secrete many, many chemicals. And many of these chemicals are known to inhibit the growth of mold and bacteria and so on. And so finding that strain or strains would be very useful.
Yeah, or even, putting that into a spray of some kind, or
yeah.
And other thing that I saw in the paper too is that, in addition to the density and the size of the buds, it was the number of leaves in the actual flower too contributed to that as well, correct?
Yeah. So this is the first time I've actually taken an entire flower apart using a pair of forceps and scalpels, sort of like a surgeon would. I took the flower and cut it up into its various components. And all growers will know that we've got these bracts, which are the sort of the inflorescence leaves that surround the center of that female flower. And really, the more inflorescence leaves you have, generally speaking, the higher your THC and CBD, because the trichomes are found in these little bracts surrounding the pistils. So I went through and literally cut them all out and started counting them. And the larger buds had more of these inflorescence leaves or bracts. Again, because it provides protection against the flower, and it increases the humidity in there. So, like you said, the bigger flowers, more total THC, more CBD, but also more of these inflorescence leaves are building up in there. So visualize a flower structure that's more open, where air can sort of filter through. It's got fewer leaves on the outside. Lower yield, definitely lower yield, but less less yeast and mold. So it's sort of a trade-off.
Right.
You don't want too small flowers. You got no yeast and mold, but then you've got no THC or CBD, so to speak.
Yeah.
Somewhere there's a happy medium.
Yeah. So, I guess in terms of, for giving practical advice to growers who are trying to lower their TYM, in terms of plant genetics, what should they be thinking? Should they be trying to select strains that, have certain morphology that are going to be more favorable for lower levels? Should they be increasing their airflow? What do you kind of advise?
Yeah, so I think the breeding part is still a ways off. I'm sure there are growers that have made correlations with certain strains are known to have higher yeast and mold. A lot of the strains, incidentally, that have high yeast and mold are also very sensitive to botrytis. So growers know which strains they grow that have more botrytis bud rot. They can stay away from those, because those are the ones that are also going to build up higher yeast and mold. Go with your botrytis tolerant strains. They would have a tendency to have lower yeast and mold, because the flower structure is much smaller, less dense. Obviously, keep the airflow happening. Lower humidity is always good for both botrytis and yeast and mold. Don't leave a lot of leaf litter on the floor.
Right.
Where the tendency is to just cut leaves and drop them. When the leaves get dried out, there's a ton of yeast and mold that naturally start to grow on that. And anything that disturbs that leaf litter on the bottom will create some of these spores, flying up to the top of the flower. Now, flowers are sticky, right? They're producing resin, and we've shown that if you have a lot of yeast and mold in the air, they'll actually land on the flower and stick. They'll stick onto that resin, and they're not killed. They're not inactivated. So those flowers, when they're taken to the lab and tested for yeast and mold, may actually have higher levels because of the fact that spores in the air were sticking to the surface of that resin. So that's something else to obviously be aware of. During harvest, don't create a lot of activity around the plants in terms of too much shaking or too much jiggling, because that'll just create a spore dust, if you wish, that will land and stick to the flowers.
Yeah, that's a good.
Yeah.
Sure. Sorry, go ahead.
So as far as spray, sometimes people say, well, can I spray Xerotol or can I spray some product that's registered? I don't think it's a good idea because you're increasing that humidity, moisture level in that flower, particularly when it's closer to harvest. So really, there's not much you can do from a treatment perspective to bring those levels down.
No, and I understand too that a lot of it isn't so much what happens during the growing of the plant, but it's in the post-harvest activities, the drying, the curing, and the trimming. I wonder if you could speak a bit about what you found around that sort of activity.
Yeah. So that's another aspect that's super important. So let's leave the grower aside. You've picked up a whole batch of harvested flower, and now you're deciding, how am I going to trim this? Do I want to do the standard wet trim, where you push all the flowers through into a trimmer, and then dry the buds, or do you want to hang dry, where everything is left intact, and then the moisture is taken out, and then you can trim off your leaves? We found a pretty big difference between hang dry and wet trim. The wet trim creates some damage, obviously, to the leaves and releases chemicals or nutrients that a lot of molds that might be present, particularly penicillium, start to feed on it and grow. So even during the drying process, you're seeing buildup of these molds in a flower that's been trimmed wet. On the other hand, when you dry this thing first, you're not allowing these wound sites or broken off leaves as a source for mold and yeast to get in. The entire flower structure is dried. You've eliminated 90% of your yeast and mold. And then when you start trimming it as a dry trim, we found the yeast and mold levels were way lower. So that's one thing that definitely will make a difference. A lot of growers are using the hang dry system. They'll probably find they don't have as much issues with yeast and mold. We didn't look at anything beyond that. In other words, the curing process or the packaging and storing process. I think all growers know that when you store inappropriate humidity or temperatures, you're going to get buildup of yeast and mold. And they know that if you cure them too long or under high moisture, you're going to get yeast and mold. So we didn't really look at that aspect. We kind of stopped right at the point where the flowers had been trimmed, whether it's wet or dry, and then collected data from that.
No, that's interesting. So do you have any idea, like in terms of percentage, what growers are typically doing? Is it more common for them to be doing wet trimming versus dry?
They're shifting a lot towards dry trim because it's less work in a sense. They can bring the flowers into the dry rooms, hang them, and then deal with them later. The drying process itself is very detrimental to yeast and mold, which is obviously a good thing. When you have dry herbs and dry products, dried figs, and they tend to last a lot longer because the moisture is taken out of them. Similarly with dried flower. The problem is that you never get down to zero. I've never seen a dried flower get down to zero mold, yeast and mold. There's always a fraction that still hangs around. And that population can always build up later on if the storage conditions aren't right. But if you're coming in with a very high yeast and mold load, then nothing you do after harvest is going to bring all of it down to zero. So there's two components. Try to reduce the yeast and mold while you're growing, and then after you harvest and bring it in, try to do things that will reduce the yeast and mold even further in that product that you're handling.
Yeah, and I have to imagine that leaf litter too would be a big consideration during the trimming process.
Yes. Yeah, and even having too many leaves on there can create problems for not enough moisture drying and so on. So hang drying has to be done carefully because that you've got way more plant material, and you want to make sure that all of it gets down to, I don't know, 12 to 14% moisture, so that you're not leaving remnants of yeast and mold even in the hang dry samples. I would
And would it
say
be,
over the year of study, like we looked at maybe, I don't know, 2 to 3,000 samples. If everything is done right, samples are not too much yeast and mold in the beginning. They're hang dried and carefully handled. In the entire batch of 2,000 samples, I would say the average yeast and mold was around 2,000. So 2,000 would be sort of the average that you would expect if everything was done right. You brought things in properly, they were hang dried and trimmed and so on and so forth. 2,000 is doable in most states and definitely doable in most countries, but never will it ever go down to zero.
yeah, no, and I was going to ask you too about you were mentioning the hang drying and there being kind of leaves and kind of creating maybe the moisture environment that's similar to what you were talking about during growing. So with that in mind, important to have good air circulation when you're doing the hang drying as well?
Totally, totally. And in fact, we actually see an explosion of yeast and mold when the plants first enter the drying room, because there's so much on there that they just start moving around. So you're right. Get the airflow happening. Make sure it's a long process, 7 days, 5 to 7 days. Get the moisture out. Measure the moisture in there to make sure it's low enough before we take it out and start trimming it for final product. There are pockets of yeast and mold in there. I have seen in some of the flowers, for some reason, there's little pockets where you're looking at 10, 15,000 CFUs per gram. But if it's dried properly, it should go down to between 2 to 5. I would say is a good number that anybody would be happy with.
Yeah, no, absolutely. And as you said, you're never going to get down to zero unless you maybe do some irradiation. I don't know what kind of experience you have with that, or if you have thoughts on that. I know it's sort of a touchy subject in the industry.
So that's allowed in Canada. We're allowed to irradiate with e-beam radiation. I have looked at samples before and after from the perspective of yeast and mold. It's pretty much zero after it's been irradiated. What does it do to the final product? People that have used it would say that it doesn't really alter too much of the cannabinoid profile. It may perhaps impact some of the terpenes, but overall they're finding not a significant difference between irradiated and not from an organoleptic perspective. The problem is the cost. It's really expensive to go through and treat all of the product unless you absolutely have to. For example, if you're exporting and it's a requirement for zero mold, then you would have to do that. But it's expensive. There's other ways, and I think the article we published was trying to address what are these other ways that could minimize your yeast and mold levels without having to resort to things like irradiation afterwards.
Yeah. And I know there's different types of irradiation. Which ones were you looking at? Was it ozone or?
No, we looked at electron beam radiation, e-beam radiation. I haven't looked at ozone. People have talked about ozone. There's also plasma, cold plasma treatments. The Israeli researchers have done good studies on that. They've published a couple of papers on different forms of radiation that are effective. And most of them do very similar results to e-beam radiation that we've looked at. But there is a cost factor, and there's also the handling factor, right? You got to then take your products, move them over to the site that's being treated, and then bring them back for packaging and so on.
Yeah, absolutely. All right, but it does seem like there's hope here that, if best practices are followed, then you should be able to hopefully get underneath the threshold as long as your state has set a realistic one.
I think so. Now, we haven't talked about outside grown, field grown.
Yeah,
that's another story altogether, because you have so little control over the weather. If you get rained on, if it's a series of hot 100-degree weather with high humidity, it's almost impossible to manage yeast and mold. That's really unfortunate. And I don't have any suggestions or recommendations there because I haven't worked with outdoor grown cannabis in terms of yeast and mold. It would be definitely more challenging to manage those plants.
Yeah, I can tell you as someone who has grown in the Northeast and had plants that finish in October, it can be very tough to manage mold.
Yeah. And you know, something just occurred to me. Maybe there should be criteria for regulations indoor cannabis versus outdoor, where there's more leniency on the indoor, because you know that the yeast and mold pressure overall is going to be lower indoors if everything's done the way it should be. Outdoors, maybe that's a different scenario. So I don't know if the regulatory agencies consider method of production or not. There could be some way to discriminate indoor or differentiate, I should say, differentiate indoor grown versus outdoors in terms of potentially having lower CFUs.
Yeah, no, that's a great point. All right, Zamir, do want to be very mindful of your time. And I already know that I'm going to be putting a link to the paper in the show description for people to check out, because there's a ton more information in there that we didn't get a chance to get to. But in addition to that, if you have any other resources or research that you'd like to share with the folks and that I can put links to, please let me know. And then, of course, any ways that people can maybe get in touch with you or follow the work that you're doing, please plug away.
Sure, I appreciate that. And again, it's an exciting field to be in because there's so much to learn. And the idea of exchanging information to me is very important. The fact that we're talking today is because I want the information to get out there. I want it to be used. You guys have a good way of disseminating it. And the more we share, the more we know, the more we understand, and the more we can manage these problems. These problems are not new. They've been around for centuries, since the day cannabis was first grown. I suspect the yeast and mold tolerance was much higher back then than it is now. But we need to know, we need to study, and we need to share.
Absolutely. No, and we appreciate you coming on again, Zamir, and I'm sure you'll be back. You'll be back again. And if we don't see you before CanMed, I can't wait to see you down in Marco Island.
Thanks a lot. Appreciate it.
All right.
Thanks, Ben.
I hope you enjoyed my conversation with Dr. Punja. Check out the links in the show description to learn more about the topics we discussed. And thanks again to this episode's sponsor, Agilent Technologies. In the meantime, please like, comment, and subscribe on whichever platform you use to listen to us. Also, please check out canmedevents.com to sign up for email alerts and learn all about our annual Innovation and Investment Summit. You can also view videos of all the past CanMed presentations in the CanMed archive at canmedevents.com. I invite you to join our CanMed community group on Facebook to interact with other cannabis professionals. And please do follow us on social media. We're on Instagram, LinkedIn, Twitter, and Facebook. Just search for CanMed Events. All right, that's it from us. Stay safe, stay healthy, and be sure to join us for the next CanMed Coffee Talk.